Okay, step number one, when you come in, wash your hands. Okay, we have 2 sinks in the room. Get in there and scrub up a little bit. You can also just wash before you come to lab, that would be fine. Just make sure your hands are clean. There's 2 reasons for that, well, maybe 3. The first reason is it's good hygiene. Yeah, we're in a pandemic, so keep yourself tidy and clean. The second and more important reason in this case is you're going to be working with fly food, and you do not want to contaminate the fly's food with fungus and mold and nasty things like that that'll grow on it. So you're basically dealing with food, so wash up. And then the third reason is that personally I don't think flies like those filthy, dirty, slimy humans getting their fingers all over the food they're going to eat. Okay, so that's number one. Just wash up before you get in here. All right, step number 2. While you're back here, in this cabinet we have all these fly bottles. So what I want you to do is grab 4 of them. These are all sterile. They've been autoclaved, so they're ready to go. Also, if you look down here, we have this little drawer, and it's got a bunch of foam stoppers, so grab 4 of those. And now we're ready to go make up some fly medium. All right, step 3, you're going to bring your bottles back to the bench. Notice that what I have done is I have put everything you need to culture flies on the bench top. So you don't even need to go back of the room to do anything. Just sit here and do it. I do have extra supplies, extra medium and so forth in the very back, and I'll show you where that is during class. Okay, so here, this is really important. Let me just shift this a little. You need to label your bottles. So yeah, there's labeling tape at the bench. I kind of recommend that if every group uses a different color tape, it will be easier to find your flies once they're all piled up in that cluttered incubator. So apparently I'm going blue or yellow here. Okay. What I want you to label these with is the kind of cross you're doing, and also the date and something to indicate who it's from. That can be your initials. You can think of some clever name for your group. Anything goes. Okay, so here, for instance, I'm going to label this bottle. This one is going to be ST by ST. That's Scarlet. And today is the 22nd of January. And I'll just put my initials there. Okay, and you're also going to label one BW by BW. So we're going to cross 2 brown-eyed flies together. And once again, the date and something to indicate your group. And then repeat that again. All right, so you're going to set up pairs of bottles here for this. Why? Because especially the first time you do this, you will screw something up. There's no shame. You're learning. You'll occasionally mess it up. So we're just doing a little redundancy here. And that will just make it more likely that we'll get all the flies we want. Now, what are we doing today? Well, we are going to culture, as I just alluded, we're going to culture scarlet-eyed flies and brown-eyed flies. And on this first run, all we're doing is generating a stock. So we're not doing any fancy cross. We're just going to be crossing flies with the same genotype with each other. And one of the things we want to do there is verify that they're actually true breeding. So in a couple weeks, you'll get to take, in less than 2 weeks, you'll get to see the progeny of this cross. And for instance, from the BW by BW bottle, they better all be brown-eyed. If they're not, something went wrong. Okay, so we got all our bottles labeled. We are now ready for cooking breakfast for flies. Step 4, let's make breakfast. So what you need is, you need your bottles, of course. I have some containers containing fly medium, these little things right here. I also have a packet of yeast at each of the bench top positions, and I have these little cups that we're going to use to measure things out. Oh, also these cheap jack funnels. So we're going to be pouring out this stuff. Try not to make a mess, and if you do make a mess, clean it up afterwards. Okay, let's get started here. So I'm just going to put that funnel there. Going to open up. Ooh, I already spilled some yeast. All right, so what do we want to do? We want to get 2 level scoops of the fly medium here. Just dump that in. You notice I'm not being real precise with this. This is an art. Okay. Put 2 scoops in there. We could get by with just 1, but I feel like we are more likely to get more uniform coverage of the medium in the bottom if we do 2. All right, close this up. Then what we're going to do is, I've got a beaker of deionized water right here. If this beaker is empty, at the back of the room there are those 2 gray taps. That's just deionized water. It's just here so that you can be really lazy and convenient and just scoop it up here. So what we're going to do here is we just get, again, 2 scoops of water. Notice the magic trick. When you add water, this particular medium turns blue. Makes it easy to see what's going on. Okay. Wasn't that tricky? Yeah, you've just made a fly maggot's breakfast. This medium is what the larvae are going to consume, but we also have to make provisions for the adults that we're going to put in there. What are they going to eat? Well, they eat yeast. So you've got your little packet of yeast. Just grab a little bit of this and count out 1, 2, 3 grains if you can. There, 3 grains. Don't overdo it. You just need a few grains of yeast, and there, that's ready to go. Now all we need to do is add flies. So I will just skip through the rest. I'm going to set the other 3 up, and I'll be right back with the next step. Okay, everyone. Next step. At each bench there is going to be a bottle of flies that I set up. So this, for instance, it says ST. These are scarlet-eyed flies. And there's the date. I set this up 10 days ago. So after 10 days, what we've got is a good population of flies in here. There are little larvae tunneling through the blue stuff. And you might also see on the side of the bottle there's some pale larvae that have crawled out. After the larvae get to a certain size, they try to get out of the medium. They crawl up the side, and then they pupate. So you see these tan-colored little rice-shaped grains here. That's the pupae. And then after they pupate for a while, they hatch out as adults. So if you could look in here, you'd see that I've got a fair number of adult flies crawling around, just buzzing around, and they're still laying eggs, so we're still getting egg production in this bottle. Okay, so each lab bench will have one of these. You're going to anesthetize these flies, and you're going to set them aside so that you can share these with the other groups. So one bench is going to be doing wild-type flies, another one's going to be doing brown-eyed flies, and another one is going to be doing scarlet flies. And you have to share. You have to look at each other's flies, because I'm going to have you compare them in a little bit. But first we have to anesthetize. We have to knock them out so we can work with them. Okay, how do we do that? Again, it's going to be really easy. So there are bottles labeled fly nap on each bench. Those are the only bottles I want you to use for anesthesia. Do not do not, under any circumstances, add fly nap to any of your good healthy bottles full of healthy flies, because the fly nap will get absorbed into the medium, and you're basically going to OD all the flies in there. So don't do that. We're going to confine it to here. So the first thing we have to do, though, is we have to transfer flies from this bottle to this bottle. Okay, how do we do that? Well, here's how I do it. Let's pop that out. I am going to then take this bottle and invert it. And notice, here's the tricky part. You want to invert it, and then you want to remove this foam plug quickly. And, oh, how quickly can I do it? I got stuck there. Oh, a couple flies got away. Get rid of those. Invert it over the bottle, and then what you can do is just gently wrap it to knock a bunch of flies down. Okay, you do not have to try and get every single fly out. We're sampling the population that's growing in this bottle. And then you want to restore the foam plugs without them escaping. You just kind of do this. Look at there. Wasn't that elegant? And then we put another plug in here on our flying net bottle. Okay, so now what I have is a bunch of scarlet-eyed flies buzzing around in here. To anesthetize them, we use this simple anesthetic called fly nap. Oh, there's a fly on it. Get away. This is fly nap. Fly nap kind of smells bad. I think it smells like the inside of an inner tube. So it's just got this kind of plasticky smell to it. But it's totally harmless to you, as far as we know. It's been tested. It won't hurt you. But it puts the flies to sleep. So what we're going to do is you just open this up and grab one of these little wands, and you slip that in there, dip it in the wand, and then pull it out. Quickly put the lid back on, because otherwise in no time at all the whole building, the whole room will smell like fly nap. Okay, and then just slide it in there. See? So now our flies are sniffing fly nap, and we'll just wait a few minutes, and they'll pass out and they'll end up on the bottom of the bottle. Again, I don't know if you can see that, but right now they're all crawling up the sides. It just takes a few minutes for them to pass out. Once they've passed out, again, you want to watch them. You don't want to overdose your flies. So once they've all kind of fallen to the bottom of the bottle and they're not going anywhere anymore, they may be sitting there twitching a little bit. It's kind of cute. Think of a little kitten taking a nap and just sort of twitching its legs. That's the way the flies are going to be there. You don't want them totally inert. You want them kind of groggy and lying around and avoiding trouble. I'm just gonna, this might take a few minutes. We'll just keep filming. You can get an idea how long it takes. I see them constantly falling off the sides. They're getting kind of drunk right now. Okay, once they've all passed out and they're all kind of lying on the bottom of the bottle, and you can help them by giving it a knock here, you're going to have maybe half an hour or more to work with them, so plenty of time. Depending on the dosage you've given them, they can sometimes stay out for an hour or two, but they won't be flying around anymore, which is what we want. If you see flies that have escaped, and they will inevitably, some of them escape, I'm going to be harsh and pitiless and say just kill them. You don't know who those flies are. You don't know where they came from. You don't want them interbreeding with your stock. Okay, right now I can tell. Yeah, they're getting pretty sluggish. A lot of them are just lying in the bottom. There's a few still walking around. I'm going to wait for them to stop walking about, and then we're going to transfer them to either a 3x5 card. Nope, this is a 4x6 card. Or we can put them in a petri dish. And I have small petri dishes around here too. What do you think? Do those look quiet enough for you? No, there's one that's still running around. Again, you don't want them to be totally inert. If you see the flies all lying there with their wings pulled straight up behind their back, that probably means you've killed them with an overdose. Yeah, there's that one. There's always one. We'll give them just a minute. Okay, I think that's good enough. So we're going to go on to the next step. I'm going to transfer these to a card. It's kind of nice to have them on a white card. It's really easy to observe them that way. And let's get them all out here. You do want to make sure you remove all of the flies from the fly net bottle so that they don't trick somebody else later on. Okay, so we've got them all on the white card. Then what you can do is, please don't try and pick them up with your fingers. That's fatal. We've got paintbrushes all over the place here. Just use a little paintbrush and you can scoop them into a tidy pile so that you can observe them on the microscope. All right, we have a couple of microscopes at each bench. We want you then to take these flies and examine them carefully. Before you start looking at them though, okay, see I got my nice little piece of paper here with all the flies lined up on it. Label it. I'm just going to put ST here so I know that these are scarlet flies. Also, if you're going to use a petri dish, and I am, get a piece of tape and put a label on it. Okay, again, it can just so that we know what kind of flies are in here. So here it is. I've just labeled this ST. Each bench is going to have its own line of flies, but you're at some point going to start sharing them with all the others, and the worst thing that could happen is if you confuse the different lines of flies, and that messes up your cross. So you want to be crystal clear always on what flies you're working with. Okay, so got a bunch of flies here. We're going to gaze upon them, and there's a couple of things you need to do. The first one is you have to learn how to tell boys from girls. There are these nice handy Carolina Biological Supply Company manuals here, and they have a section where they show you the difference between males and females. So here's the sexing section, and what you can see right away is they differ in their abdomens, and there's another feature that we can use to tell them apart. Okay, so I'm going to go to the bigger microscope so we can see where we can all see this. So you're gonna identify the sex. You're gonna separate males and females. Okay, gang, here we are at the microscope of my lab, which just happens to have a nice camera attached to it so I can record this. And what you're seeing here is a pair of wild-type fruit flies. And here I've got both a male and a female in view. How do you tell them apart? Well, look at the female, and you will see it's got kind of a stripy appearance to the abdomen. So it's just a series of stripes going down the abdomen. Black stripes, thin black stripes. When you look at the male, you see a large prominent black blob at the end of the abdomen. So that's a quick way to just detect right away who's who here. You can just scan through there. This is not entirely reliable, however, because particularly right after they eclose, right after they come out of the pupal case, they tend to be very pale, and so the stripes don't show up very well. So what you should always do is confirm whether you've got a male or not, and to do that, we're going to look at the front legs. So even at this power, I hope you can see, there's a little dark area on the male's forelimbs. This is something called the sex cone. Only males have it. And what I like to think of is it kind of looks like elbow patches on its front limb. So let's zoom in a little bit so we can see that a little more clearly. Focus. Okay, let me shift this just a little bit. There's our male. And look at that front leg. And what you can see is that there are some dark bristles right there. These are used in their courtship and grooming and all that kind of good stuff. Only the males have them. Just look for that. Also, what you can see as you look at these guys is, oh, hey, they're looking pretty healthy, pretty happy. They're sound asleep, but they're doing that little kitty paw twitching, as you can see. So these are flies that I'm confident will do just fine if I put them into a culture dish. Unfortunately, these flies are destined to go into my spider cages, but hey, live food, it's delicious. And then what you're also going to do is look closely at the eye color. This is a cross where we're going to be looking at eye color, and that can be subtle. So these flies are scarlet-eyed. They have bright red eyes. We also have flies that have brown eyes. They're dull brown. And then there's wild-type flies which have kind of brick red eyes. You need to look at these and figure out who is who, because you're going to have to score the progeny of the cross later on. Okay, so the other thing you need to be comfortable with is identifying these mutant strains. So what I've got here is 3 flies. As you can tell, they are female flies. And one of them is scarlet-eyed, one of them is a wild-type brick red eye, and the other one is a brown-eyed fly. So get used to looking at those colors. I know they're a little bit subtle, but after you stare at them for a while, you'll get comfortable with them. You have to be able to score a fly's eye color even if you don't know what it is, if I haven't told you what it is. So let's zoom in just a little bit more. Focus. I hope these show up well in the recording. Yes, the brown-eyed fly is kind of twitchy right now. Yeah, the scarlet-eyed fly is just kind of sticking its tongue out. The quietest of them is the wild-type fly. Alright, so now you know how to tell a boy fly from a girl fly, and you know how to tell a brown-eyed fly from a scarlet-eyed fly from a wild-type-eyed fly. You've got brown-eyed flies cultured, you've got scarlet-eyed flies cultured. We're going to grow those up so that you have lots of flies to work with, and then eventually what we're going to end up doing is doing a cross between brown-eyed and scarlet-eyed flies. What color do you think the eyes of the progeny will be? Make a prediction. Actually, next week I will go through in detail and explain to you exactly what we expect to see. So that's for next week. Okay, at the end of the lab, behold the fruits of your labor. You're going to have 2 bottles of scarlet-eyed flies and 2 bottles of brown-eyed flies. You are not going to be culturing the wild-type flies. That's just for you to look at and compare with these other mutant strains. And then what you're going to do is take those and back of the room. No, actually the front of the room. This is our incubator. Second shelf is for the Tuesday section. So just put yours in there, and you can see why using your own colors will be helpful, because it's going to get cluttered in here. What are these doing in here? Those are ancient. We'll get rid of those. So you're just going to set those, and you're going to leave them alone for about a week and a half. What I recommend is that you do come in on fly time. That is, come in after they've been cultured for a few days and check on them. Make sure that the medium looks good, that it's blue, it's not covered with mold, anything like that, that you've got larvae emerging, you've got pupae, then you're golden. If you don't, if there's something wrong, contact me and I'll come running up. We'll figure out what's going on. And it's, as you see, it's easy. You can start another one. I'll knock out some more flies for you. Okay, last step, of course, is you're all done. Clean up your mess. So look at there. I got some medium here. I got paper towels. There's a sink over there. Just make sure you clean it up for the next group to come in. All right, that's it. So we'll see you next week.